Hi everybody, let us discuss some of the questions from transfer RNA and translation of molecular basis of inheritance. Let us directly enter into the first question. Who postulated that the presence of an adapter molecule that would on one hand read the code, on the other hand would bind to specific amino acids? Nirenberg. No. Nirenberg cracked the genetic code. FSA Crick. Yes, he only postulated. J.D. Watson. No. He, along with the Crick, proposed the double heliced model for DNA. Morris Wilkins. So he, along with the Rosalind Franklin, gave the X-ray diffraction data of DNA. So second one is the correct answer. Coming to the answer two. Coming to solutions. Crick postulated that the presence of an adapter molecule that would on one hand read the code, on the other hand would bind to specific amino acids. Nirenberg deciphered the genetic code. J.D. Watson, along with the Crick, proposed the model for DNA in the form of double helix. Morris Wilkins provided X-ray diffraction data of DNA along with Rosalind Franklin. Coming to the next question. Imagine behaves as adapter during translation. That is transfer RNA or soluble RNA. A3. Cellular factory for protein synthesis. This is ribosomes. So B4. C has UTRs. UTR means untranslated regions. Messenger RNA. Binds to stop codon, release factor. The release factors can bind to a stop codon. Only releasing factor 1, releasing factor 2 can bind to stop codons. Not releasing factor 3. So D1. So A3, B4, C2, D1. First option must be the correct one. Let us see the answer. So transfer RNA behaves as adapter during translation. Ribosomes, cellular factory for protein synthesis. Messenger RNA has UTRs. Release factor binds to stop codon. Going to the next question. During charging or amino acylation of transfer RNA, amino acid is bound to. So during charging of transfer RNA, amino acid bound to 3' end of acceptor end of transfer RNA. Not 5' and 3' end. That is CCA end or amino acid attachment site. So there, amino acid is attached to the last nucleotide known as adenine nucleotide or adenosine monophosphate to the second or third carbon of the ribose sugar. The amino acid is attached by esterification. So third option must be the correct one. Not DHU loop of transfer RNA, anticodon loop of transfer RNA, no. 5' end of acceptor end of transfer RNA. No, it is 3' end of acceptor end of transfer RNA. Third option must be the correct one. Let us see the answer. During charging or amino acylation of transfer RNA, amino acid is bound to 3' end of its acceptor end. It is attached to the ribose sugar of last nucleotide, that is adenosine monophosphate, at either second or third carbon by formation of ester bond. They formed between carboxylic group and OH group. Carboxylic group of amino acid, OH group of the sugar present at the second or third carbon. The end, that 3' end, is called as CCA end or amino acid attachments. This is the 3' end. Coming to the next question. Two statements are given. The codon on messenger RNA is 5' AGU 3'. Is read by transfer RNA with anticodon as 5' to 3' as ACU. ACU. Absolutely true, because the codon and anticodon are anti-parallel and complementary. It is 5' to 3' AGU, the codon, then the anticodon must be from 5' to 3', means opposite to uracil of the codon on messenger RNA, adenine comes. 5' adenine comes. Next, GC, AU, it's correct only, statement 1. Transfer RNA with anticodon as 5' ACU, 5' ACU 3' carries the amino acid serine. Absolutely true. Serine has got 6 codons. So out of 6 codons, the two codons are present along with the arginine. The two codons for serine are ACU and ACC. Right? The other four codons for arginine are UCU, UCC, UCA, UCG. So first statement correct, second statement is also correct. The transfer RNA with anticodon as 5' anticodon 5' ACU. It carries the amino acid serine. Right? So AGU, AGC. AGU, AGC are the codons for serine. Corresponding anticodon will be for AGU, it is ACU only from 5' to 3'. Right? Besides AG, it also has that codon AGC, another codon. Right? Transfer RNA with anticodon 5', 3' ACU carries the one answer absolutely. Statement 1 and statement 2 are correct. Fourth option must be the correct one, yes. Codon and anticodon are antiparallel. The codon 5' AGU 3' of messenger RNA is read by anticodon ACU from 5' to 3'. Present on messenger RNA. AGU, AGC, UCU, UCC, UCA, UCG are the codons for amino acid serine. The transfer RNA with anticodon 5' ACU 3' carries the amino acid serine. Right? Be very careful here, codon and anticodon. Coming to the next question. In which are the following stages ATP are utilized? ATP are utilized during the charging of transfer RNA. During charging of transfer RNA, the ATP are utilized. Binding of messenger RNA to ribosome. No, ATP are not used. Binding of initiated transfer RNA to first codon or messenger RNA. Along with the transfer RNA, even GTP is also associated, but GTP is not utilized, associated but not utilized. Binding of release factor to stop codon. Binding of release factor to stop codon. No utilization of any ATP. Only during chain termination, GTP are utilized for dissociating various components of protein synthesizing machinery. Charging of transfer RNA, absolutely true. Fourth option must be the correct one. Let us see the answer. ATP are used during charging of transfer RNA. ATP are not used during binding of messenger RNA to ribosome. Association with GTP, initiated transfer RNA is bound to codon or messenger RNA during chain initiation. At this time, there is no usage of GTP. GTP are delayed while dissociating the various initiation factors. GDP and release factor are bound to stop codon during chain termination. Absolutely. GDP plus releasing factor 3 are bound to stop codon. Coming to the next question. Assertion reason. Initiated transfer RNA that carries methionine is different from non-initiated transfer RNAs that carries amino acid methionine. Yes, absolutely true. Initial transfer RNA is different. It is given even in the book. But in what way it is different, the reason is not given. Just he has given the different. Absolutely true. It is different only. In anticodon's time, the initiated transfer RNA has 3 successive GC pairs with hydrogen bonds between them, but not in non-initiated transfer RNA. In non-initiated transfer, there may be 1 GC pair, but not 3 successive GC pairs. This is the main difference because of which the initial transfer RNA can occupy the P site of the ribosome. And P site and A site are distributed in both the larger subunit and smaller subunit. Right. Assertion is correct. Reason is also correct. The reason clearly explains the assertion. So A and R are correct, and R is the correct explanation of A. Answer to. Initial transfer RNA has 3 successive GC pairs in anticodon step. In non-initiated methionine transfer RNA, it has CG, AU, and GC, not consecutively GC. Initiated transfer RNA cannot bind to A site of ribosome and binds to only P site. P means peptidyl site, A for amino acid site. Non-initiated first binds to A site and later shifted to P site during translocation after formation of peptide bond. Coming to the next question. Arrange the following events of translation in correct sequence. Binding of initiator amino acid transfer RNA to the initiating codon of messenger RNA. This is done only after binding of messenger RNA to smaller subunit. Catalytic activity of 23S ribosomal RNA. It's seen only during chain elongation, but A is the chain initiation event. C. Binding of messenger RNA to smaller subunit of ribosome. This is chain initiation initial event. This. So C is first, after that A. So after that, catalytic activity of 23S ribosomal RNA, absolutely. So its catalytic activity peptide bond is formed between the two amino acids. After the formation of peptide bond only, there is movement of ribosome a codon distance towards 3' end of messenger RNA. So A and C are related to chain initiation, B and D are related to chain elongation. In A and C, the first event is binding of messenger RNA to smaller subunit of ribosome. Next event is binding of initiator amino acid transfer RNA to the initiating codon of messenger RNA. So C first, A next. After that, catalytic activity of 23S ribosomal RNA in chain elongations. B. So C, A, B, and last one is D. Movement of ribosomes a codon distance towards 3' end of messenger RNA. First option must be the correct one. Let us see the answer. The various events during translation are: number one, binding of messenger RNA to smaller subunit, binding of initial transfer RNA to smaller subunit, messenger RNA complex, binding of larger subunit to form initiation complex. So this is also initiation. Binding of second amino acid transfer RNA at A site of ribosome to the codon with its anticodon. This is the first step in elongation. Peptide bond formation between the adjacent amino acid. Second step in chain elongation. Movement of ribosome a codon distance toward 3' end of messenger RNA. This is the third step, which is also known as translocation. So repeatedly there is translocation. The peptide bond formation takes place repeatedly, right? And the translocation also takes place. The last stage is chain termination. Coming to the next question. This one, binding of release factor to stop codon and separation of polypeptide, messenger RNA and ribosomal subunits and transfer RNA. Coming to the next one. Identify the correct statements. Ribosomes move each time a codon distance during elongation phase of translocation. This is correct only. About 80 different proteins are present in ATS ribosome. This is also correct. I think last time it was given in one of the NEET exams. 80 different proteins are present in ATS ribosome. It was given. Stop codons are recognized by release factors. Absolutely true. The stop codons UAA and UAG are recognized by releasing factor 1. UAA and UGA are recognized by releasing factor 2. And releasing factor 1 and 2 are in return recognized by releasing factor 3. UTRs are messenger RNA required for efficient translation process. Absolutely true. For efficient translation, UTRs are required on messenger RNA. The energy for peptide bond formation is ultimately provided by ATP. Absolutely true. At the time of charging of transfer RNA, ATP are utilized. When the charged transfer RNAs come closer, there is a formation of peptide bond between the amino acids that they carry. So all are correct. A, B, C, D, E are correct. A, B, C, D, E. All are correct. A, B, C, D, and E. Second option must be the correct one. During chain elongation, the ribosomes move a codon distance each time towards 3' end of messenger RNA. ATS ribosomes have about 80 different proteins. Release factors recognize the stop codons. UTRs, one present prior to initiating codon and another one after stop codon, are needed for efficient translation. ATP is the source of energy utilized during charging of transfer RNA for peptide bond formation. The picture is given. So what is asking? Identify the biological process depicted in the adjacent figure. What is this? This shows translation process. Ribosome is there, messenger RNA is there. So some transfer RNA, peptidyl transfer RNA, one of the amino acid transfer RNA attached to ribosome. Another amino acid transfer RNA carrying the aspergine. ASN means aspergine. It is about to enter onto the surface of ribosome. So another one transfer RNA is leaving the ribosome. So It is in the process of chain elongation of translation. The translation. Charging of transfer RNA, it is translation 3. Let's see the answer 3. The chain elongation process of translation, because one transfer RNA is leaving from ribosome, the peptidyl transfer RNA is at one binding site P site, that is of larger subunit. Amino acid transfer RNA is at another binding site A site, and another amino acid transfer RNA is ready to participate in translation machinery complex. So about to enter aspergin carrying the transfer RNA. Aspergin transfer RNA. Coming to the next question. Which amino acid is carried by a transfer RNA whose anticodon is GUA? So GUA is the anticodon, means codon must be UAC, right? The corresponding codon for this anticodon, we read from 5' to 3' means the 5' base of the codon must be the complementary to the 3' base of anticodon. That is complementary to A is U. For U, A, UA, for G, C, UAC. So UAC is the codon for tyrosine. UAC, codon for tyrosine. Tyrosine also has got another one, UAU. UAU, UAC are the codons for tyrosine. Must be tyrosine. Tryptophan, UGG is the codon. Right? UGG is the codon for tryptophan. Right? For cysteine, UGU and UGC. UGU, UGC for serine, we know UCA, UCC, UCG, UCU, right? These are the some of the codons of serine. So answer must be 2. The anticodon is 5' to 3' GUA. The corresponding codon messenger RNA must be UAC. As codon and anticodon are anti-parallel and complementary. Codon UAC indicates the amino acid tyrosine. Tyrosine also has another codon UAU. Please like, comment, subscribe and share. Thank you.